Welcome, listeners, to Talking Eyes. I'm your host, Leanne Trin, an optometrist and public health clinician based in Larrakia Country, also known as Darwin in the Northern Territory of Australia. This podcast was created to translate exciting, innovative developments from vision research into a form that both clinicians and really any curious mind can understand. Innovation, simplicity, clarity. This series will be diving into a topic where the research has been absolutely awe-inspiring: the retina. It's just a small part of the human eye, roughly the size of a postage stamp, but it's an incredible piece of human biology that plays a central role in how we experience the world around us. Bionic eye technology represents a promising avenue for restoring vision in individuals with profound vision loss to improve quality of life and independence. A number of research institutions and companies in Australia and indeed globally are actively engaged in developing bionic eye devices and conducting clinical trials to evaluate safety and efficacy. I have with me today Associate Professor Penelope Allen, who is a Principal Investigator at the Centre for Eye Research Australia. She's the head of the Bionic Eye and Vitreoretinal Research there, and also head of the Vitreoretinal Surgical Unit at the Royal Victorian Eye and Ear Hospital. Welcome, Penny.
Oh, thanks, Leanne. Lovely to be here.
Penny, I know you completed your medical degree back in the mid-80s. I can't imagine there being many female medical registrars or ophthalmologists at that time. And you've said in the past that specialising to become a vitreoretinal surgeon was not deemed particularly female friendly. Why ophthalmology for you? Why vitreoretinal unit? And why bionic eye? It seems like you have a personality keen on challenges.
When I started training after finishing medical school, there weren't many women in surgery, which was how I then got into ophthalmology via surgical training at the Royal Melbourne. I found ophthalmology interesting because it seemed like it was this niche area that a lot of people didn't really know anything about, and using the instrumentation to examine the eye was a skill that you needed to develop, but it actually interacted with a whole lot of different areas. When I was exposed to the surgery as a junior trainee at the Royal Melbourne, the eye unit at that stage was led by Dick Galbraith, and he was a very enthusiastic teacher. We were always trying to promote eye health, in all of the patients that were admitted to the hospital, as well as looking after the patients who came to the clinic. And the surgery's lovely. Then when I got into the registrar training program, I realised that retinal surgery was just so beneficial for patients. And then I did some further training in vitreoretinal surgery at Moorfields in London. So I was the first female vitreoretinal trainee in Australia. But when I went to London, then I met other women who were also working in the area. And I was very fortunate just to have a husband who was very supportive. Charles' mother was an anaesthetist, and his father was an ophthalmologist. So he was, his mother actually did more after hours work than his father. Mm. So he was always, very supportive.
Yeah, that's wonderful. Let's move on to the focus of today's episode, bionics and prosthetics. The idea of a bionic eye is to restore vision to people with very poor vision, people who are legally blind. Those who may be reliant on high-powered magnifying lenses to optimise the very little existing vision they may have. People who use braille and adaptive technology for reading and communication. And people with such significant peripheral vision loss that they require a white cane or guide dog in order to remain mobile and safely navigate their environment. In April 2008, The Prime Minister of Australia at the time, Kevin Rudd, convened the Australia 2020 Summit at Parliament House to help shape a long-term strategy for the nation's future. A bionic eye was suggested as a follow-on from the success of the cochlear implant, representing Australia's next bioengineering aspirational goal. There had already been some pre-existing work done at the University of New South Wales, and Penny was intimately involved in the early process. She recalls some of the events of the day.
Originally, Mark McComb and myself were asked to offer some advice to a team at the University of New South Wales.
So Mark McComb is another retinal surgeon.
Vitreoretinal surgeon
Yep.
at the Royal Victorian Eye and Ear Hospital. And Professor Hugh Taylor had been contacted by the team at the University of New South Wales who were working on what they regarded as an intraocular electrode, prototype bionic eye. So we started offering some advice about the devices and a potential surgical approach. And then the 2020 summit occurred, and I've never known who suggested a bionic eye at the 2020 summit, but one of the conclusions from the 2020 summit was that the development of a bionic eye would be an aspirational goal for Australia.
A collaboration formed between clinicians and researchers from a number of groups. The Centre for Eye Research Australia, the Bionics Institute, the University of New South Wales and the University of Melbourne, and NIC ICT Australia. As a joint collaboration, they were successful in their application for funding for a bionic eye project from the Australian Research Council.
Well, we started with a prototype device that the team at the University of New South Wales developed. And at that same time, the collaboration had also the engineers at the University of Melbourne were very interested in these crystalline diamond devices which were electrically conductive. They had electrically conductive cores and could potentially offer many electrodes to stimulate the retina. And hence it was thought that they could potentially offer sort of higher acuity vision. Because we had the original University of New South Wales device and we had started placing it in the suprachoroidal space, we're talking about a potential space between the sclera, which is the white wall of the eye, and the choroid, which is the blood vessel layer immediately beneath the retina.
The blood supply to the eye. And that actually, you're not talking about the implant actually directly contacting the neural retina, are you?
No.
You're talking about a space which is really close to, and we sort of covered this in our earlier introductory episode to the retina, but a space which is really close to the retinal pigment epithelium.
Yes.
And what should be a photoreceptor layer there.
So this seemed like a very good potential space because it is easy to slide the device into this space. And although originally we were very concerned about the risk of hemorrhage because it was close to the blood vessel layer, the reality is that people who have advanced retinitis pigmentosa have a very thin atrophic choroid. They have lost the photoreceptors due to the genetic abnormality that they have within their photoreceptors. So they don't have that huge metabolic drive and they don't have a huge blood flow to that part of the retina. And so, in fact, it is even easier to slide the device into this space, and the risk of bleeding is actually very low.
There's sort of two areas of focus which were sort of identified back then as potential areas of focus that the Bionic Eye project could take. One was that we could try to develop a device which was what we think of as a high acuity device, and that sort of device would really target people with issues around central vision. So for diseases of the macula like Stargardt's or macular degeneration. And then the other type of device that people thought could be potentially a good device for people with vision issues was a sort of a wide field vision device, which would enable people to have some sort of peripheral vision again, perhaps people with glaucoma or retinitis pigmentosa. Where are we now in terms of device development?
The high acuity device essentially is a very rigid device, and we were unable to solve the surgical challenges with that.
Yeah. And so the surgical challenges being it was not flexible, it didn't really adhere to that space?
Well, it could not be placed in the suprachoroidal space.
Yeah.
It had to be placed in what we call the epiretinal position, which is where certainly some of the research groups overseas have placed their devices.
Yeah. And that position is just behind the vitreous in front of the neural retina. Is that correct?
Well, you remove all of the vitreous.
Oh, you have to remove the vitreous. So the vitreous is that gel inside the eye.
Yes. So you have to remove the vitreous, and then you need to find some way to place this device in position against the retina. The advantage being it's close to the neural retina, but you do need to stabilise it in some way, and that way of stabilising it is with a tack, and it's very hard to get even apposition of the device against the neural retina, and just the fact of tacking it has a destructive element as well. There's no innate stability because it's within a space that is fluid-filled.
So there were just too many risks and challenges involved with implanting a high acuity device, essentially.
We were unable to make it stable in the eye. The suprachoroidal device that we developed actually came about because we had a lot of challenges with the device from the University of New South Wales. Once again, from a surgical point of view, from the size of the exit point from the eye. And so myself and Mark McComb and Jonathan Yeo collaborated with engineers from the Bionics Institute, and we developed a device that had initially 22 electrodes, which was the prototype device. It had a smaller exit from the eye, and we then moved on to a 44-channel device, which we used in our second clinical trial.
Yep. The 22 electrodes, how does that compare to that initial high acuity device?
Oh, well, the initial high acuity device, they wanted to have sort of 500 to 1,000, but Retina AG in Germany developed a device that had many small electrodes, and I think it was about 1,024. But the functional outcomes from that device were no better than the device from Second Sight or even from our 44-channel device. There's so much retinal degenerative change and retinal reorganisation in patients with retinitis pigmentosa that you can't just increase the number of electrodes. Expect to have small
increased resolution, greater pixelation.
No, it just doesn't work like that.
It doesn't work that way. Creating a device that would restore high acuity vision had many surgical and visual challenges to overcome. Maintaining stability within the epiretinal space, the risks that come with a vitrectomy where the entire vitreous gel inside the eyeball is removed, figuring out the exit points from the eyeball, and then navigating the existing degeneration and reorganisation of the retina. A different type of a device with a different purpose needed to be developed if there was going to be any success had with a bionic eye. Penny tells us about the new goalposts and discusses the type of eyes suitable for this suprachoroidal prosthesis.
With a device such as this, we were aiming to aid navigation, object detection. So we were aiming for people that had lost navigational vision, but still had perception of light, because we wanted to know that the optic nerve was still functional. We didn't want to put a device in a participant and then find that their optic nerve didn't work and that we'd put them through the surgery for no possible benefit.
So these people who were chosen for the clinical trial had retinitis pigmentosa.
Yes.
And it was important that they had a functional retina somewhat. And is it important that their inner retinal layer was still functioning?
Yes.
Do people with glaucoma perhaps who have peripheral field loss as well, would they be suitable for an implant?
No, because they've lost ganglion cells and they've got optic nerve abnormality. So no, we were only looking at patients with inherited retinal disease. So they had photoreceptor loss, but they had residual neuronal retina, and that neuronal retina could then be stimulated with the device. And then obviously that stimulation, the phosphines generated via the optic nerve back to the occipital cortex.
Yeah. So I think that's a really important point to make about this implant, is that we do need to have some form of existing neuronal retina intact. And the reason that RP patients are really suitable is because they have lost retinal pigment epithelium and photoreceptors only. Penny clearly has a great respect and admiration for the individuals who committed to take part in the clinical trials for the Bionic Eye. These incredible people were willing to risk their eyeballs for the unknown to become, less unknown, and commit a significant part of their daily lives to this research also.
We wanted to recruit 3, and we did for the prototype trial. And the people that participate in these trials are extraordinary individuals. They firstly have to have a surgery, and undergo the risks of that surgery. With the prototype trial, we didn't really know what the device would produce, whether it would produce any visual impulses at all or visual perceptions. Sometimes they are people that have tried other types of trials, but they are all very interested in providing data to help the next generation of people with inherited retinal disease. And the reality is that some of these run in a dominant fashion in a family, and so they may have relatives themselves who also have the same condition, but they're just very altruistic people who want to help the community of people with inherited retinal disease. The time commitment for the trial was intensive, and so they had to not only have the surgery, but then undergo training and then assessment, which we originally planned over 12 months. But because things were going well, we actually extended that prototype trial for another year, and all 3 of the participants were happy to continue for another year.
Yeah, we spoke earlier about the time commitment needed to participate in clinical trials and how difficult it can be for somebody with severe vision impairment, particularly if they're working. And a lot of these inherited retinal diseases are affecting people in their working age. Yes,
that's exactly right. And so it has to be a participant who can contribute that time. And I think the important thing for listeners to realise is that the evaluation of the function of the device it's important for that to be, done in a randomised way so that we can actually demonstrate categorically that there is improvement or there is not improvement. We have to test it properly for the results of the clinical trial to be valid and to be published.
And in terms of implanting for trials, people who participated, am I correct in saying this, that they were told that the implant would be inserted for the trial, but then would need to be removed post-trial? Is that correct? Or
No, we didn't. We did not need to remove it.
Oh, you didn't need to remove it? Okay.
No, we removed the prototype trial had a connector through the skin because it was designed as a lab-based device. So that connector was removed, but the device, it was never felt that we would need to remove it from the eye. One of our first 3 participants has had it removed from the eye, but that was for a completely different health issue. It was causing artefact on radiological investigations, which impaired their management.
And these are some of the implications of having a bionic implant inserted into your body that we hope will be there forever,
yes,
for perpetuity, is the, future ramifications of any further testing that you might need done, for example, MRIs.
Yes. And, for that reason, then these devices have to have some level of MRI compatibility, but it depends on, in a way similar to a cochlear implant.
Let's talk about the bionic technology itself, because from what I understand, there's 3 major components to the bionic eye. There's this implant, which goes into, in your case, the suprachoroidal space of the eyeball. And then there's an external camera, and it's connected to a visual processing unit. And my logical brain, for some reason, thought that the bionic eye would work really similarly to the retina. So it would replace the photoreceptors and receive light signals directly, and then, transfer through to a visual processing unit of some sort before it was transferred to the brain. But it doesn't work that way, does it?
No, it doesn't work that way.
Completely not.
No, because at the moment we don't have a way of replacing the photoreceptors. There's obviously research going on looking at, stem cells and
yes.
But for this device, we are using the electrodes within the device with low-level electrical stimulation to stimulate the residual neurons, the neural elements of the retina, and then those impulses are carried via the optic nerve back to the brain. But the camera, which is on the glasses, captures the visual scene, and then that is transferred to a vision processing unit that the patients carry. The vision processing unit turns that into electrical stimulation, and then that is passed across the skin as it's attached to the receiver stimulators that are implanted beneath the skin. So that stimulation goes to the receiver stimulators above the ear. There's a lead that goes to the eye, electrical stimulation to those neuronal elements, and then back to the brain. And then to the brain.
So it's glasses, visual processing unit, implant electrode, and then brain. So completely opposite of what I actually initially thought. In terms of the surgery and what it involves for the patient, I was actually watching an interview that involved yourself, Matt Pito, and one of the research participants, Colleen, who mentioned that she had understood that there would be a retinal implant, but the shaved head was quite a surprise to her. So if we go into what's involved for a person who participates in the clinical trial and the surgery, what are they going to expect?
Well, the actual device is the array that's placed within the eye, but then it has a lead to the receiver stimulator units. Which we used 2 in the second trial, the 44-channel trial. And so there needed to be a wound for those to be placed, and that's
an exit point essentially for those leads. Or an entry point.
An entry point, a wound to place them and then close that wound.
Yes.
The actual device was we dissected forward and then made the wound in the eye and then placed. So the lead runs between the eye and the receiver stimulators.
So is that underneath the skin or is there sort of
the
bone
muscle.
being removed or anything like that? Any extra holes that people need to know about?
There's some modifications to the surgery going forward.
Yeah.
But one of the things that we did do was stabilise the lead at the orbital margin, the bony corner of the eye here. And we collaborated with Robert Briggs, Associate Professor Robert Briggs, who's an ENT, otolaryngologist. We designed the lead so that it would sit in that corner so that it was stabilised in relation to the eye. And that's been a really successful part of the device because it has not moved in any of the participants.
Yeah, and movement's something that a lot of people don't realise how much eye movements we actually do. I know you had a room full of the skulls simulating eye movements that I saw, which looked quite freaky.
That was, and that's the thing, our eyes constantly move.
They do.
And that is the huge challenge with having an electrode array, either sitting on the surface of the retina, either beneath the retina or in the suprachoroidal space, because the eye is constantly moving.
And it's not just, regular intentional eye movements that we're making, but the eye performs these little eye movements called microsaccades,
yes,
which is really important for visual perception and to prevent neural adaptation that can happen that, changes our eyesight. You guys have really had a huge challenge to try to create a device that has long-term viability, but also doesn't move in the eye when the eye
That's exactly
naturally
right.
does these things.
Yeah, so it has to be stabilised, at the exit from the wall of the eye. It's stabilised at the orbital margin, but it's also in that potential space in a stable fashion. We can't have reliable perception if the electrodes are moving, against the residual neurons.
So to date, you've run 2 clinical trials, really incredible milestones in themselves. The first, you implanted 3 people with the device. The second, you implanted 4.
That's correct.
Leading up to these clinical trials, first of all, there was a lot of work done to make sure that it was safe, it was biocompatible, and we've just talked about how it needs to be able to withstand those frequent eye movements and be functional. You would have had to have worked with engineers.
Yes, the engineers from the Bionics Institute, and you. So you mentioned biocompatibility. So Professor Rob Shepherd always emphasised that we would develop the device with materials that were previously tested. So silicon and platinum, so that, we have known...
Really inert materials.
Yeah, known materials. You mentioned the skulls in the cupboard. The skulls in the cupboard were generating information about the leads and lead wire breakage, and trying to generate data so that we could confidently say that the devices would last, 20 years in the eye without breakages. And that's why we set up rigs with the device and the lead and electrically rotating plastic eyes that just went, 24 hours a day.
Penny had an incredible and timely opportunity to test her surgical approach of inserting the electrode into the suprachoroidal space of a live human eye. And this allowed her to understand the feasibility of the approach, whether it was possible and whether it was safe.
So we used cadaver eyes that had, been donated for various things to trial the the surgery. However, there was a patient of one of our colleagues who was having both eyes removed, and she said to her surgeon, "Is there something that I could do for research?" And he contacted us and said, I have this patient, she's coming up, she's offered to do something. And I said, well, would be very useful for us to have 2 dummy devices and just do the intraocular component of the surgery, that is, place the devices in the suprachoroidal space in a live patient as opposed to just a cadaver eye.
So when you say dummy device, this device was not
activated.
Activated, right.
No, it was not activated. So it was a...
Could it be activated?
Well, no, because it wasn't designed to be activated.
Right.
It was designed to test the surgical approach.
Right.
I spoke to her and I explained to her that it would mean that her surgery would take longer, and she said, well, that's fine. I'll be under general anaesthetic anyway, and so I'll be asleep. I'm not really that worried. And so this was a very, very useful step for us because it really was step from cadaver into, even though it was a dummy device, but it's, the same measurements, the same size, and the flexibility to go into the suprachoroidal space and just prove that our surgical approach was in fact feasible in a live patient.
Yeah, and it was useful that she also had retinitis pigmentosa also.
Very, very severe.
Yeah, so you could almost see what the outcome of an implant would do to the retinal tissue.
Yes, we could, and we did, take images of that, and were able to prove that it was in the correct position, and that the devices were sitting in a good position.
Yeah, and stable.
Well, for that period of time.
Because of this, the first prototype clinical trial progressed with greater certainty, knowing that the surgical approach was safe, able to be positioned where it needed to be in the suprachoroidal space, and importantly, stable.
So the aim of the prototype trial, that was a proof of concept trial to prove that if we did provide stimulation within the suprachoroidal space, that we would be able to generate phosphenes, these artificial visual impulses from the electrical stimulation.
Were there eye movements happening while she was asleep or no?
No.
Okay, so the eye movements completely stop when
with
people
a general
are
anaesthetic.
Right, okay. That first clinical trial you conducted in 2012
to 2014.
To 2014, and that included recruitment of your participants, and then what happens?
Surgery, and then we allow the patients obviously to recover from the surgery, and we have...
How long does recovery take?
Well, it's about a month, really.
And what does that involve for the person who has had the implant? Are they in a lot of pain, or are they...
We kept them in hospital for about 5 days. They had some antibiotics, first of all, eye drops, and then they had some skin stitches and staples, which obviously needed to be removed, but we just wanted them to be comfortable, and they were comfortable when we then moved to what we called switch on, which was when we first stimulated the electrodes to see whether there would be any responses. And it was funny, I was actually much more nervous about switch on day than I was about the surgery, because I'd had this German ophthalmologist who worked with one of the other groups, and he said, you know this approach will never work. And I said to him, well, why would we do it if we thought it wouldn't work? I mean, that just doesn't make sense. All of these participants have had no useful visual impulses for, more than 15 years, sometimes 20 years. And so...
so their vision prior to the surgery...
was perception of light. And so we were all obviously thrilled when our first participant said, yes, she, could see something with stimulation. But because they haven't had useful visual impulses for so many years, then it does take training to get used to what these visual impulses are. And also we need to determine what level of electrical stimulation is required for each electrode. So that the actual training is quite arduous because we have to determine thresholds for each electrode, and then that data is fed into the vision processing so that when we're trying to convert an image into electrical stimulation, it has to be individualised for each participant.
Being such a novel device, a huge part of Penny's research into the bionic eye has been to develop effectiveness measures that would be able to be standardised and implemented worldwide. Measures like the device's ability to detect changes in the direction of light movement, and then moving on from that micro level of vision to actually seeing whether it translated into different navigational tasks being able to be performed based on the visual information.
We use a number of different tasks for assessment because we need to, randomly test with device on compared to device off to show that there is actual, functional improvement. So we use some computer-based testing, which, is bars of light passing both horizontally and vertically at different speeds to detect whether the participants can judge where that movement is, actually being able to identify a square on a computer screen.
Were all those things possible on switch on day?
No, not switch on day. Switch on day is just detecting thresholds. This is going into training in the lead up for assessment over the 2 years of the trial. We then found 2 out of the 3 participants were doing well on the computer and lab-based testing. We thought it was worth trialling them with some navigational tasks. And so navigational tasks are setting up obstacles in an obstacle course and, being able to navigate around the obstacles. In the second trial, we used some other testing as well, identifying, a door, a high contrast door randomly placed within a testing room, what we call tabletop, which has positions with different high contrast objects. And so the participant is asked to point to the object and potentially identify that. So all of that data and these forms of testing have been used by other groups overseas because we want to have a benchmark to compare devices across the testing.
Exciting. The results of the first trial found that the devices were stable and all of the implanted electrodes remained functional during the 2 years of that first trial.
So the fact that, all 3 patients perceived phosphines and that there was some functional benefit from that array led us to develop an array with more electrodes because the feedback from those patients was that a wider area of stimulation would be useful.
So is that a larger device then?
No, same footprint.
Okay.
Because
just more electrodes.
Because just more electrodes in the same footprint.
Right.
Because I was very reluctant to change the size of the device because I felt that it could lead to further surgical issues, that, we safely implanted this device. It was a good size. We could put more electrodes on that same footprint and be safe.
Yep. And so it then leads you to your second clinical trial based on all of that feedback, the positive outcomes from that initial clinical trial.
Yes.
What sort of upgrades did you make for the second clinical trial?
The electrodes were a little larger. They were stabilised slightly differently in the array.
They were larger because
we wanted to lower the electrical impedance. We wanted to potentially make the thresholds lower,
right?
All in the interests of safety. We had a better idea of what levels of electrical stimulation were required now because we'd had the prototype study, and we also had implantable receiver stimulators so that the information from the vision processing unit could be transmitted across the skin, with the magnet attachments in the same way that occurs with a cochlear implant.
Because the first device was...
Well, it wasn't really. It was a lab-based device, so it was a computer and sitting in a lab.
Right.
But to do the mobility, we put the computer in a backpack.
Right.
Yeah.
Okay, so it was quite large. It wasn't really...
It wasn't
something that...
No, because it was a lab. It was developed for the lab.
Yes. So the second device is now you're thinking more about portability and how this device can be used
at home.
In the day-to-day.
So the hypothesis was that if the device could be used at home, that it would be more functional.
The second prototype device was a more portable device. It had glasses with a camera which fed a video image into a small vision processing unit that could be strapped around the person's waist or neck. And the unit converted this video into electrical signals to feed the electrode implant at the retina. The visual processing unit, as you'll hear, is where all the magic happens. Here, Professor Nick Barnes, a computer engineer from the Australian National University, and Bionic Vision Technologies became much more involved to develop the algorithms that communicate to the electrode array, effectively translating the video signals detected by the glasses.
If you have an object, then you need to stimulate the electrodes, in a certain way, so that you can perceive edges, perceive objects.
So really, because generally your visual environment has so many details to it. So there's a challenge there, isn't there?
Oh, it's a huge challenge. Yeah. It's much, much more challenging than a cochlear implant for the inner ear because there's so many more neurons involved in the retina. Nick has also developed some algorithms for depth, for proximity to objects, and then Bionic Vision Technologies, which is the company that was developed from Bionic Vision Australia to commercialise the device, have also developed a couple of other algorithms because interviews with the participants identified some software modifications that would be helpful. So, for example, one of our participants would be able to perceive your shape. Now, we know we're sitting at a table and that you're a person, but if this was in the street, could it be a bollard? Could it be a small tree? Is it a person? So a face detection algorithm is very helpful because it can determine that there's a face. Identifying an empty chair in a cafe or at a table or in church, it can be very helpful because it enables, once again, social interaction. And what we would really like this device to do is aid navigation and social interaction and object recognition.
Over the following 2 years, the second clinical trial with the updated electrode array was conducted. The electrode was the same size, but hoping to achieve a wider field of vision with a greater number of electrodes, moving from 22 to 44 electrodes. The safety profile, stability and effect on existing ocular structures was monitored, and ways to measure the performance of the implant were better established.
During the 2 years of the trial, the participants were assessed. I mentioned some of those lab-based tests, which we continued to do. But the functional test
around mobility, obstacles, identification.
Identification of, doorways in a randomised fashion.
Mm-hmm.
So there's always sort of like 3 positions, starting positions, and, the door is placed randomly in different positions, and it's a high contrast, and it is identifying where that high contrast door is and how close you get.
Yeah.
Yeah.
And so those tests are really great in terms of research, because you can make those tests uniform across different sites. So somebody overseas can use the same test to demonstrate. But then in terms of real world practicalities, not everything is high contrast, obviously.
No. And so we used a tool called the FLORA, which is the Functional Low Vision Rated Observer Rated Assessment. And that was developed by Second Sight, a company in the US who developed an epiretinal device. And so this is essentially a collection of tests that are done in the participant's environment, be it at home or outside. And you can run the tests before the device is inserted, and then you run them sequentially, baseline over 2 years. And you can assess how hard or easy it is to do a particular test, and whether the device aids in doing those particular tests, be it sorting, washing. So, for example, sorting, darks from lights, then obviously prior to insertion of the device, then none of our participants would be able to sort dark washing from white washing. But using the high contrast algorithm, then they can do that.
Yeah, and that's a great example of a test, because it doesn't rely on learned experience for sorting washing, does it? You're not sort of navigating the bookshelf that you've known is there. It's actually purely a visual task.
Yes. And look, the FLORA is not, as an assessment task, is not completely applicable in Australia. And in fact, my team at the Centre for Eye Research Australia, Lisa Lombardi and Lauren Masalam, have been actually developing a tool that is more applicable in Australian conditions, once again, to be able to be used not only for bionic eye trials, but in assessment of people with profound vision loss, and potentially for other trials as well, potential gene therapy trials.
What sort of things aren't applicable?
Well, we don't have a lot of sort of zebra crossings, particularly where our participants lived. There were a lot of items on the FLORA. It took a long time to do. There just were some items that were more applicable to daily life.
Yep.
That things that we all need to do.
And so the outcome of the second clinical trial then
was that it was the devices, the surgery was safe. We were able to place them in position reliably. They are stable in position. Adverse events were, as major adverse events did not occur. And then the functional outcomes showed that all of those forms of testing were better with device on compared to device off. And if you looked at the results of the FLORA, there were certain areas that became easier with the device on.
And so you would have some idea of what the real life impact was for some of these people as well. And I when I was watching that interview with Colleen, it's obviously the part that brings everyone joy who works on this project to see the actual impact on people of the device. What do people like Colleen see when they're using the device? And are they actually learning more and more as they are in the device for longer and longer? Are they seeing more?
They learnt more and then plateaued. COVID came and it severely impacted our interaction with the participants, and we wondered whether that would mean that their function dropped off. It didn't though, because but they weren't able to use it as often because we were all limited in what we were doing. They weren't going out and doing lots of things because none of us were. But
so up until COVID, were you interacting?
Oh, regularly.
Yes. Regularly
that was part of the trial. So
would they come in,
yes,
see you,
and we would go
be in
to
the research lab,
and we would go to them.
Yeah,
and that was all the 3 monthly assessments for the 2 years, so that we have all that data, and then we can look at those results so that it, we have valid results.
Were there assessments that you could conduct remotely, or were there?
Well, we could talk to them about, whether they were using the device and that it was still functional, but we can't objectively assess what they're doing over the phone, just as we can't objectively assess most eye patients over the phone. Every time they do an obstacle course, for example, they're timed. Every object is randomly placed at strategic intervals. If they brush a device, it's all documented, and that's how we analyse it for the papers. So
there's a huge amount of setup involved before somebody has to navigate these tests, and so it's not just a matter of setting up a webcam and watching them in their home.
No, the results have been positive.
Many of us take our vision for granted and don't realise that from the moment we are born, capturing focused light information onto the retina is just one component of eyesight. So much of our vision and visual behaviour is actually a learned experience. As Penny mentioned earlier, higher resolution images by increasing the number of electrodes does not necessarily equate to better visual performance. What's the feedback from different people who've had these implants? What do they have to say about these implants?
Well, they would like it to work better. Better means better visual discrimination, being able to truly navigate independently, because at the moment they can't truly navigate
and is that
independently.
Something that is software related, or we spoke about earlier about that visual processing unit now having different buttons on it depending on the visual environment that this person is experiencing?
Subsequent to the second trial, then we have done work on depth algorithms. So the software, so 2 cameras instead of 1 camera, so on the glasses, so that there is a brighter phosphene as you come closer to an object, because depth perception, proximity is very important as far as not bumping into something. And face detection, empty chair detection, so that we can then have, as you mentioned, the latest vision processing unit, which has these different algorithms that can be swapped between.
The Bionic Eye concept in Australia was kicked into gear by a significant government grant over a decade ago, and that's allowed all this research and development to progress to this day. Where are we now? How far is regulatory approval for a device, or how far is a device from commercialisation being on the market?
We applied to the FDA for what we call breakthrough recognition, which we achieved. It means that the FDA sort of understand that the concept is good. They don't have enough data to actually give approval because we only had 4 patients in our second trial.
How much data is needed for approval?
It would need more than 20 patients, the majority of which to get FDA approval would need to be done in the US. And so we would have a site in Melbourne, multiple sites in the US, and the company at the moment are working on obtaining funding to run that sort of multi-centre trial.
So what are the long-term challenges, you think, to commercialisation?
The long-term challenges, there are the market has always been smaller than that for a cochlear implant. There are many more children who are born with hearing impairment compared to profound vision loss. And there has obviously been a lot of interest in gene therapy recently. The issue with gene therapy is that an individual mutation needs to be addressed to try to intervene in each form of RP. I think that running a large multi-centre trial with a device like this, is obviously very expensive. And
are we very collaborative globally, or is there a lot of competition?
Well, the other groups overseas have chosen different surgical approaches, so it's not really quite possible to be sort of collaborative in that sense. The Japanese have quite a similar approach to us in that they have gone from externally but created a big flap within the sclera, the wall of the eye, and placed their device in the flap. So they're sort of similar to us. The German approach was subretinal, which is a very complex surgery. You need to actually create a retinal detachment and place the device in the subretinal space. And Second Sight in the US placed their device on the surface of the retina in the epiretinal position.
So we're still a few years off then, really, because we need to develop these multi-centre trials.
Yes.
Get regulatory approval.
And have the results of that to submit to the FDA to get regulatory approval. And then if there was FDA approval, then presumably the TGA would then approve as well.
Yeah. Look, I think the Bionic Eye has come a long way since that initial injection of government funding from the Australian Research Council. And there's a lot of potential, isn't there, for advancement in software. You've obviously developed a fantastic technique of insertion for the implant, and the implant itself is obviously all amazing achievements. I know when I speak to people with RP, they want things to progress faster.
Oh, don't we all? Don't we all?
But, we need to conduct these studies to make sure that it's safe, to make sure that it is as effective as we want it to be, and that it does provide benefit to people. But hopefully when we get there, it'll be a real fantastic tool for, as part of a whole suite of tools that are being developed concurrently for vision restoration and vision optimization.
And it might be that a couple of different approaches are the way to go, and I suppose the thing that we're very pleased about our approach is that it doesn't preclude anyone from having some of the other approaches as well.
Yeah. So how do our listeners now stay informed about participating potentially in bionic eye research if they do have vision impairment, or even clinicians who might think that they could refer a patient to you to be part of a clinical trial, whether they're here or overseas in the United States? How would they stay informed?
The Centre for Eye Research Australia now has a much larger database of people with inherited retinal disease, the Venture database. It doesn't mean that you have to participate in anything. It's a way of being informed of what is available with your particular form of visual impairment.
So signing up doesn't mean that they will be part of clinical trials or anything like that. They just purely can stop at staying informed or participate if they wish.
If they wish. But it means that they will be informed about what's available, and then they can decide in what way they want to participate, whether they're, and, to a certain extent, even, collecting a lot of information about the progression of disease is really important.
It is.
We need to know how diseases progress, to know how many people might need an intervention. And yes, looking at the Centre for Eye Research Australia website, there's always a lot of information there about various trials that are going on. If people are interested in research, then it makes for interesting reading. Our 7 participants are incredibly altruistic people who have wanted to contribute to the research, to get it further, to make it better for people in the future. And, we can never thank them enough.
Everyone, not just you, everyone, I think. Everyone who works in the eye space and wants to offer the people who see us with these blinding eye conditions, particularly the inherited retinal diseases, which affect people at a really young age.
Yes, exactly.
An option. And so being part of research really moves our progress in understanding and learning, and then hopefully managing conditions in the future.
That's exactly right, Leanne.
Thank you so much, Penny, for joining us. I have learnt so much from you today, and I am really so overjoyed that people like you are working in this space and have people who are willing to participate and advance this field of science for people. Vision restoration was quite science fiction-y, quite futuristic, and now it's here. It's so exciting, isn't it? So thank you so much, Penny. Thanks for listening to Talking Eyes. Additional resources for this episode can be found in the show notes. If you found the content of our podcast interesting, feel free to post a great review and share it with your community. If you have a topic you'd like to hear more about, or have expertise in the field of vision and would like to contribute to future series, contact me via our socials on Facebook and Instagram. I'm Leanne Trin, your host of Talking Eyes.